ahead and introduce the speaker briefly, and then we can start the talk. Dr. Rajan was born and raised in India, a country with diverse cultures, religions, traditions, and languages, all nearby his home. Thus, he appreciated diversity from a young age. After obtaining his BS in Biological Sciences in 2007 from the University of Lucknow, he enrolled in a doctoral program at Sanjay Gandhi Postgraduate Institute of Medical Sciences, one of the best medical schools in India. After gaining extensive experience in the gastrointestinal motility field during his doctoral studies, he did postgraduate research in the same field by joining the lab of Professors Rowe and Sanders, leaders in the field of gut motility research. From 2021, Dr. Rajan became an assistant professor in the Department of Physiology and Cell Biology, School of Medicine, University of Nevada in the US. He has mentored first-generation graduate students and fueled their aspiration to become successful in academia by training them with adequate skill sets and future research goals. So thank you so much, Dr. Rajan, for joining us today. Let's start the talk.
Again, good morning, everyone, and thank you, Mina, for the nice introduction. And I want to thank a lot Professor Elaine Yamaguchi for providing me the opportunity to talk on this event. And I would like to thank the Women Chemist Committee at the American Chemical Society for organizing for such wonderful meeting. And very nice to meeting you all with the esteemed scientists also in the breakout time. So I want to introduce myself that I'm a researcher in the field of neurogastroenterology and motility, and learning and understanding more about the causes present in the disorders of the gut-brain interaction, like the gastroparesis, functional dyspepsia, slow transit constipation, and love to share our discoveries to the people with worldwide. So it's truly honor to be here, and I'm going to share about my journey so far building as a biomedical scientist. Here is my disclosure. So working as the board member of directors and the scientific advisor at the Rossview Therapeutics. So here is the outline I'll be discussing today's talk. So starting with my brief introduction of my scientific journey in India and in USA, and I will talk about the burden and pathophysiology of gastroparesis and RNA therapeutics, and why it is important to fix the real pathology present with the gut dysmotility and diabetes, like why the cellular remodeling. So there is a KIT positive cells, that is receptor tyrosine kinases. They are very important. So there is one cell in the gut, so that is known as the interstitial cells of Cajal, that express the KIT, that tyrosine kinases. And KIT signaling is very important for their function. And ICC has been regarded as the main pacemaker activity in our gut, so that maintains our GI motility. And there are some cells in the pancreas also. So those are known as the pancreatic progenitor cells. They are also KIT positive, and they are the main source for the producing the pancreatic beta cells. So I will talk about those cells. And also recently, there has been studies on the muscularis macrophages. So they are tissue resident macrophages. So they are very important for the gut motility and diabetes. And in the last slide, so I will talk about the intrachromophilin cells and concludes the further perspectives. And you can stop me anywhere and love to take any question and make it like more interactive. So I have split my talk in two parts. So in the first part, I will share my experience from India, where I pursued my doctoral program under the guidance of Professor Uday Ghoshal. And I will talk about the challenges I faced and the skills I developed and the opportunities I have to work with some of the best clinical scientists in the field. So in the second half of my talk, so I will talk about my journey in the US as a postdoctoral researcher and also faculty member at the University of Nevada, Reno, School of Medicine. I will share my experience working with Professor Seungil and Kenton Sanders, who are the leader and one of the best gut motility researchers in USA. And I will share my insights on how this experience has saved my career and helped me to grow as a researcher and as a person also. So I hope by sharing my journey, I will be able to shed light on opportunities that biomedical scientists have in clinical environment and the challenges they may face along the way. So after completing my master's in biotechnology from University of Lucknow in 2009, so I got more insights working in biomedical research field. So I qualified one exam that is known as the Indian Council of Medical Research. So that allows me to get the fellowship during the doctoral program and medical research institute in India. So I was lucky enough. So I got like the SGPGI, that is one of the best medical institute as me in India. And I was fortunate to get Professor Uday Gosal lab in the GI physiology and pathophysiology in Department of Gastrointestinal. That was the first lab in India stabilizing the GI physiology and pathophysiology and doing the manometry studies. And this was a dream come true for me. And I was excited to work in a clinical environment where I could combine my passion for a biomedical researcher, like the interact with the patients and then doing some basic research. So it transformed me to have to do like the translation research. So this was the start of my journey in the field of biomedical sciences. So I want to share some experiences like what I have learned during the doctoral program in clinical environment. So I had the privilege of working on both basic and clinical research projects, like which gave me the well-rounded understanding of the field. And the experiences of being part of clinical environment allowed me to bridge the gap between the lab and the bedside, as I was able to directly observe the impact of my research on patients' lives. And in biomedical setup, you have the opportunity to publish the clinical papers and also the translation research paper both. So that's the very good opportunity I got. And I have worked in the field of functional gastrointestinal disorder. So that's defined as the disorders of gut-brain interactions. To underscore the term functional, because people think that, oh, you have a functional problem, you have everything in the brain, you don't have problem in the GI, but that's not true. You have the real problem in your gut. So you have to treat that disease. Don't doctors just say like you have the problem in the brain only. So that's not true. That's why it has been reconsidered and defined as a disorder of the gut and brain interaction. So in the gastroenterology clinic, so like the 50-60% of the patients, you will see they are like with the DGBI. So like they have or functional dyspepsia, irritable bowel syndrome, gastroparesis, or slow transit constipation. And there is a huge burden of this disease, like both in the community. So I have done studies in communities also, and also in the clinics. So I found like the 60-70% of the patients are with the DGBI. And during my doctoral program, there was scanned data about the prevalence and pathology present with these diseases. So I got opportunity to work on the clinical projects like the epidemiology work and the genetic components, what are the predisposing for the functional dyspepsia, and also the clinical characterization of the patients with IBS, achalasia, and gastroesophageal reflux disease. So one very important thing I want to emphasize that interaction and connections with patients teach you a lot about the symptoms interpretation and connecting the thoughts underlying pathology present with the disease. So so what is the tool for that? So how you can connect with the patients? So we implement a proforma. So proforma is like the setup of some questionnaires. Usually doctor collect the clinical history from the patients for diagnosis of for a disease condition. So we developed a proforma. There was current criteria that was around 3 criteria for functional dyspepsia. So we use that for the patients with functional dyspepsia. Also, we use that for the epidemiological study. So it was in English, then we translated in the Hindi language. So, and we validated in the in patients and also in the community subjects. So it was like a privilege for participating in a groundbreaking study on the prevalence of functional dyspepsia in rural northern Indian population. So we did a huge study. So it was like around 5,000 people involved in the study, and we found like the overall the FGID like that include the functional dyspepsia and also include the IBS. So total prevalence was around 21.7, and only dyspepsia was only 14.9, and there was IBS dyspepsia overlap patients that was around 4.1. So, and I got opportunity to work closely with some medical resident, and it's very important to collaborate with your colleagues or in the department, what they are doing. So you will get more insight and help you to work more and also publish more clinical papers. So I collaborate with them and I was able to learn hands-on experience with cutting edge techniques like high resolution esophageal manometry, 24-hour pH impedance monitoring. So which helped me to accurately diagnose the patients with achalasia, because esophageal manometry is important to know the esophageal motility. And also I told about like lower esophageal sphincter, how tight is that or how loose is that. That is very important because whatever like you have the acid in the stomach, so it can reflux to your esophagus. So if your lower esophageal sphincter is not tight, not enough tight, so your acid will come and you have the reflux esophagitis. Or if it is very tight, so it is also not good. That is in case in the achalasia where you eat food, so food will be just jumping in your esophagus. So that will cross the esophagus and food will not go in the stomach. So patients have a lot symptoms like the chest pain, heartburn in case of the achalasia and also in case of the GERD. So they are totally opposite. In GERD, LES is relaxed a lot. That's why acid is coming in esophagus, and in achalasia is very tight, so food is not going in the stomach. So this collaboration led to several clinical publications I showed here. So that allowed me to meaningful contributions to the field and expand my expertise in this area. So one more thing is very important in our field, like we need to get regular publications. Is very critical for advancing our career in biomedical scientist. So people will know better about your contribution in the field, and also you will be recognized more within the scientific community. And also you have the better chance, then you will get more hired by further career steps. So I will share my experience and insights, my time in the United States, and what I have learned from working with such extreme leader in the field, like Professor Seungil and Kenton Sanders. So after gaining extensive experience in the field of mortality from India, so I was ready to take the advanced research. So like I want to know what's exactly going on at the cell level. So I joined this Professor Seungil lab and Professor Ken Sanders as a postdoc fellow, and to pursue my gut mortality research. So this was the major milestone in my career and allowed me to further expand my horizons and contribute to the field in a new and innovative ways. So again, I will say having a strong publication record is very important. Not only does it showcase your expertise and contribution in the field, but it also opens numerous opportunities. And in United States, a good publication record can significantly increase your chances to receive the funding, especially like in form of NIH like R1 grant, or if you are in postdoc duration, so you can apply for the K99 grant, so that you can pursue your career independent. Also, if you have good publication record, you can have like the outstanding visa, like I earned because I published like 3 gastroenterology paper during my postdoc period. Those are like the top journals in the field. And based on that, I got my own visa also in the field of neurogastroenterology and mortality. So as I said, like these are the key publications I contributed, and I will share some remarkable studies I have done so far in USA. So we talk about a lot about gastroparesis. So how it is defined, gastroparesis? So it is the gut motility disorder and is defined by the patients with delayed gastric emptying in absence of any mechanical obstruction. So in the presence of some symptoms, like patients have nausea, throwing up, satiety, bloating, lack of hunger, heartburn. Sometimes they have the reflux also. So, but in terms of etiology, the majority of the patients are idiopathic. So you don't know what's going on wrong with them. And 50% they are like the diabetic. So we know that diabetes is causing the problem. And some patients, like the 5%, they have the post-surgical or post-infectious. And the gold standard for diagnosis for the gastroparesis is the delayed gastric emptying. And that is considered delayed if the food is in the stomach for more than 60% at 2 hours, and the scan at 4 hours more than 10%. So how it is impact the life and what's epidemiology exactly? So the prevalence is not exactly known till now still, but 9.6% patients per 100,000 men and 37.8 patients per 100,000 women. So it is very common in the females, 4 times common, and it significantly negatively impact the patient's quality of life and on the health and care system. And one important etiology I mentioned, like the diabetes, so which accounted like for almost 1/3 of all cases of gastroparesis. So so I want to here say like because the 50% there is overlap, so I want to talk about what we know about diabetes. So it is common worldwide. So this map is showing that the prevalence is currently like 425 million people worldwide have the diabetes condition, and it is increased like the 48% by 2045. So huge trend there. And what about in US? So in US, like 30 million people are living with diabetes, and 84 million are at the risk of having diabetes. So either they are underdiagnosed or they are like the pre-diagnosed condition. So I like this image, like the 1 in 3 adults have pre-diabetes. So it could be you, your dog walker, or your cat jogger. So one of the major manifestation of diabetes is the GI complications. So as you can see here, the diabetes effect across the GI tract. So it can reduce the motility on the esophagus region, and also like leading to heartburn and chest pain. And diabetes can affect the gastroduodenal motility. So you feel the delayed gastric emptying, and that is hallmark for the gastroparesis. And also diabetes impairs the small intestine and colon motility. So very often patients have the constipation, or sometimes they have the diarrhea symptoms also. But most common feature is the constipation they get and gastroparesis. So so when we eat food, so this cartoon is showing that when we eat food, so there is fundus region. So the food is accommodated in the fundus. And then there is pacemaker activity, so in the carpus region, so that generates the slow waves, and then food moves towards the antrum region. So there is the pressure pump and the peristaltic pump. So they have to coordinate it. So if they're not coordinated, so the patients feel the pain in the stomach, and also they feel the nausea symptoms. And the food is in, if there is hypomotility in the antrum, then food is in the antrum for a long time. So they feel the abdominal pain. So what happened in the patients with the gastroparesis? So in the normal cases, so I have shown here like in normal cases, the like liquid food or like the solid, they should be cleared within like the 4 to 6 hours. But in patients with gastroparesis, it stays in the stomach for more than 10 hours. So that's the reason they have all the symptoms. So, sorry, practically what we have learned on the cellular pathologies that we learn about gastroparesis, they are almost based on the deep tissue biopsy studies, and that have yielded important clinical insights in pathophysiology. So as you can see here, this is the stomach wall. So, and the deeper tissue of the stomach wall, there is the muscularis propria, and most of the studies have been done in the antrum and gastric corpus, and also pylorus region. And in the summary, what we have learned, there is a loss of c-KIT. So that's why I was talking about that. KIT signaling is very important for ICC. And the doctors have seen that these ICCs, they are lost. And they were correlated with the delayed gastric emptyings in patients with diabetic gastroparesis. And these studies also support like there are some neuronal marker, like the pan neuronal marker, like PGP, there are some neurons very important for maintaining the gut motility. They also lost in patients with gastroparesis. And there is one more important cell is known as PDGFR alpha positive cells. So they are electrically active, and they have the purinergic transmission, and they coupled with ICC and smooth muscle cell that create the SIP syncytium. And SIP syncytium is very important maintaining the gut motor functions. And there are recent studies, they showed like there is loss of CD206 positive macrophages. So they are resident macrophages in the stomach. And if these macrophages are lost, so they correlate with the loss of the ICCs. And that's why the patients, they have the gastric emptying delayed. So ICCs we learn here, and then macrophages. So they are like the pacemaking cells in the GI. So what we believe, like a disease is caused by there is some alternations in the inner functioning of the cells. So it is very critical to understand how healthy cell works, and more important, like we need to understand what happens when disease strikes. So one of the important regulatory mechanisms mediating the mechanisms are like the microRNAs. So they play important role in gut health. And they are very small, tiny molecules, like 21 nucleotide bases, and they mediate the post-transcriptional gene expression by inhibiting the translation, and they are master regulators of the cell differentiation, proliferation, and apoptosis. And this was discovered by Andrew Fire and Craig Mello, so they won the Nobel Prize in 2006, and they published this study in the Nature in 1998. So this was the milestone study. They showed how these microRNA, they repress the protein synthesis. So as you can see here, like there are some cells. Those cells, if these microRNA are enriched, sometimes so they are functional. If they're lost, so they are not functional. So, and sometimes some microRNA, if they're deficient, they also lose the function. So depending on which microRNA is enriched and which microRNA is lost. So this regulation regulate the key pathological mechanisms in gut motility disorders, not only GI motility, but also the immune function, intestinal barrier function, and also the visceral sensation. And these studies like provided us impetus, like exploring the microRNA dysregulation in these cells and what might be the potential pathogenic factor leading to dysfunction of these cells, and through specific gene target regulation in diabetes, GI dysmotility. So to identify abnormally expressed microRNA, so we want to know in ICCs particularly what microRNA are expressed and in a healthy condition, and what microRNA they are lost in diabetic condition. So that is diabetic and dysmotility condition. So we created a mouse line in our lab, so that is known as the KIT-COP GFP lab overview mice. So we can track the ICC in diabetic condition based on the marker KIT that is important for the ICC. And we got performed the microRNA sequencing analysis, and you can see the top 10 microRNA. So there was one microRNA that is known as the microRNA 10B that was enriched in the ICCs of the healthy colon, and also they are enriched in the jejunum, but they are decreased. So green color is the decreased expression of the microRNA, and red is the increased expression of the microRNA. So you see, like in diabetes condition, they are totally reduced, and also and in the diabetes condition, they are reduced and they are enriched in the healthy conditions. So we found like only one microRNA. So that was so exciting for us, like is highly expressed in ICC and in healthy mice, but it was drastically depleted in ICC from diabetic mice. So for the determination of the functional role of the microRNA 10B, we generated the KIT-restricted micro-10B knockout mice. So we just deleted one microRNA, tiny microRNA in ICC, and then see that mouse may develop the diabetes or gut dysmotility or not. So what we found that in mice where the microRNA 10B are deficient in ICC, so there was ICC degeneration. So it means that ICC lose their functions, and they develop the gut dysmotility and diabetes. So as you can see here, the analysis of the immunohistochemistry. So by the confocal imaging, So in the 10B knockout mice, so knockout means like we deleted the microRNA 10B in ICCs in the tissues of the whole GI tract. So in vitro, like we did the delayed gastric emptying tested, like that is hallmark for the gastroparesis, and we found that these knockout mice they have the gastroparesis and they have the slow transit constipation phenotypes. So slow transit constipation is like mostly we did the colon transit time delayed, and also the fecal pellet output was reduced. So further, we also did the glucose tolerance test. So as you can see in the red line, so that is the micro-10B knockout mice. So they have the impaired glucose tolerance, and also they have the impaired insulin sensitivity. So both are the hallmark for confirming the diabetes phenotype. And these mice developed the diabetes and the gut dysmotility. So not only like we have done in the murine model, we also validated everything in the human patient samples. So these are the patient data, like the patients with diabetic gastroparesis and also patients with um, idiopathic gastroparesis. And we found similar pattern in patients with diabetic gastroparesis and idiopathic gastroparesis. Micro-10B levels was reduced, and it was intact in the healthy subjects. So what are the possible microRNA treatment approaches then? So it may include either like the introducing the microRNA inhibitors or microRNA mimics. So to rescue the cell damage and to restore the functional activity of the damaged cells in patients with GI dysmotility. So there are like the microRNA mimics and microRNA inhibitors. So microRNA mimics may be useful where a particular microRNA is deficient, and microRNA inhibitor, so where there is functional loss if there is very high expression of the microRNAs. So good part is that these microRNAs are naturally occurring, so molecules in our body, and they have the very less side effect because we are not using any chemical reagents to treat these disorders. So we asked the question like in these mice who has like the diabetes and also the GI dysmotility, can we, if we restore these microRNA in the cells, can we restore the diabetic and gut dysmotility phenotypes? So we compared the treatment outcome in micro-10B mimic and anti-diabetic and prokinetic treatment at post-injection 4 weeks and 8 weeks. So for the anti-diabetic medication, we have like the liraglutide, sitagliptin, insulin, and for the prokinetic, we use the prochloride. At micro-10B, we did 2 injections, first week, like the 0 week, and at the second week. And then we compared all the clinical outcomes at 4 weeks and 8 weeks. So what we found that the glucose and insulin tolerance, they were significantly improved in micro-10B mimic injected mice. And anti-diabetic, as you compare with the anti-diabetic, so they were sufficient to reduce the diabetic phenotypes. Only the medication was on till 4 weeks. But when medication was off, so the diabetic phenotypes, they come back again. Same as the insulin and also the glucose tolerance test. And also we found like the similar thing happened with the GI motility phenotypes. So there is one very good prokinetic medicine, there's no prochloride, so that restored the GI dysmotility phenotypes. But we found that these medicines, when they're on, only till that time they're effective. When they're off, so then again the mice came back to the original pathology like the GI dysmotility. So it signifies that these medicines are not fixing the real problem. So they're not remodeling the cells, but the micro-10B, they remodel the ICCs and also the pancreatic beta cells. So our like the murine, metabolic, and also the GI motility data that evidence the profound and prolonged efficacy of the micro-10B compared to the anti-diabetic and prokinetic medications. So as you can see here, as I said, that micro-10B mimic injections likely stores the proper functioning of the ICCs and beta cells by increasing the expression of the KIT and insulin in micro-10B knockout and high-fat, high-sucrose male mice. And that leads to the reversal of the GI dysmotility and the diabetes. So we published this paper in gastroenterology in 2021, and we used like state-of-the-art methods, and we explored the mechanisms and how the microRNA dysregulation in ICC's beta cells that lead to the diabetic gastroparesis and cellular constipations. And we found that if we deplete the microRNA 10B, so that leads to the gastroparesis and cellular constipation and diabetes. But if you restore these microRNA, so you can reverse these disease states. And it could be very important, clinically beneficial for the patients with diabetic gastroparesis. So I want to talk about another cell that is known as the macrophages. So recent studies have noted that if there is loss of the gut resident macrophages, so they are like very important for the gut motility, and they interact to the integumentins and ICCs and smooth muscle cell for maintaining the motility. And what we have learned that I already mentioned in previous slide, that if you lose there is macrophages known as CD206 positive macrophages. So they are like the more anti-inflammatory type. So if you lose those cells, so there is the diabetic gastroparesis, and they also correlate with the loss of the ICCs. And this is data from the Mayo Clinic group at Rochester, and they found that CD206 positive macrophages, they are lost in 90% patients with diabetic gastroparesis. So we want to see that how the microRNA 10B mimic is impacting the muscularis macrophages population. And we use like the high fat, high sucrose diet. We fed mice like 4 to 6 months. They become diabetic, and also they have the delayed gut motility. And we did the flow cytometry, and using some stem cell markers for the macrophages. And then we characterize them for the pro-inflammatory macrophages and for the anti-inflammatory macrophages. For the anti-inflammatory, we use the marker CD206, and that is important for the human patients also. And what we found that in high-fat, high-sucrose fed mice, the pro-inflammatory macrophages, they were very high population. But in the mice, when we injected the 10B, so this population was reduced. So there was less inflammation. And you can see here, like for the anti-inflammatory, like CD206 positive macrophages population, it was increased in the mice injected with the microRNA 10B. Also, we confirmed the micro 10B and how it can restore the cytokine profile in diabetic gut. So we did like this is ELISA data only for the cytokine profile in the pro-inflammatory cytokines and anti-inflammatory cytokines. So these cytokines profile was restored in the mice injected with the microRNA 10B. So one another important pathophysiology for these disorders are like the impaired epithelial barrier function. So that is also known as the leaky gut phenotype. So as you can see here in the healthy and disease condition, how the barrier integrity regulates the gut motility and immune functions. So in healthy conditions, you will have like the intact enterocytes. So there is some tight junctions. So they maintain the epithelium. So there is not the leakage in the epithelial barrier. So when it's maintained, so gut lumen, so there are some bacteria, food antigens, so they will not cross the barrier. But in disease condition, what happens? So their enterocytes are damaged, and there are some tight junction proteins, particularly Jo1 protein, and these are the pathogenic bacteria. They cross the barrier and they reach to the ICC or immune cells, particularly the macrophages. So there are more inflammation. So then we found that what is happening in the micro 10B knockout mice. So we found that these mice have also the impaired intestinal permeability, and this leaky gut phenotype was mediating the whole gut transit time studies and also mediating the diabetic phenotypes. But when we injected these micro 10B knockout mice with 10B mimic, we found that when we restore the microRNA, so they are restoring the proper functioning of ICC, beta cells, enterocytes, and macrophages. And the last slides, I want to show one other important cell that is known as the enterochromaffin cells. So these are the cells which are the major source for the serotonin in our body. and they play very important role like the gut physiological processes like the motility, absorption, secretion, chemosensation, or mechanosensation. So these cells have like the apical surface towards the gut lumen, and also the basolateral surface where like the all the your muscles cells are there like ICC, macrophages, and the PDGFR cells, and how they connect with the brain also. So they maintain the gut-brain axis. So in brief summary, I want to conclude one study we did. So we have one mouse model. So we can deplete the intrachromophene cells. And what we found that when we deplete the intrachromophene cells, so these mice have the delayed gastric emptying. And also they have the colonic dysmotility. But when we supply the serotonin, so all these like the disease parameter, they were rescued in these mice. And this paper also we published in gastroenterology. And not only in the mouse model, we confirmed these cells depletion in patients with idiopathic gastroparesis. And in idiopathic gastroparesis, they also have the EC cell, they have the less EC cell number, and they have the less serotonin content. And that was like the negatively correlated with the gastric emptying at 2 hours and 4 hours. So in this study, we reported like a potential mechanistic link between the serotonin deficiency and delayed gastric emptying. So this illuminate the pathogenic mechanisms that may underlie the gastroparesis more generally. So there are lots of clinical trials going on, but most of those they are failed. So we think that for the next steps, we should conduct the clinical trials that focuses on targeting the pathophysiological mechanisms, such as like the intestinal barrier function or gut motility. So fixing the cells are more important, not just treat the symptoms, but to fix the real problem. And also is very important, like the food we eat, the gut microbiota we have, and how the food is handled by the host and microbes, and the immune response of the host to the luminal content, and how all the GI events and how they are sensed by the brain. So this is my future perspective, like the DGBI research area is evolving and understanding the heterogeneity of pathologies at single cell level and spatial transcriptomics of like the ICCs, interneurons, macrophages. So they will open the window for potential therapeutic options. And exploring also the gut microbiota and their metabolites and how they crosstalk with the host, and also gut to the brain, and how they can remodel the gut-brain axis from motility to mood. And the modern approaches like the integrating the multi-omic data from the multicentric longitudinal studies that might be also exploited. And also the more understanding of the pathologies and fixing the cellular defects linked with the pathophysiological mechanisms, that would be the best therapeutic approach for treating disease and all these like the gastroparesis and slow tonic constipations. So I strongly believe like the connect the dots of the past learning. So what we have learned, like the doesn't matter like chemistry background or like you are from the biology background, but that will help you in your future. So you have to construct the foundation, what you have learned today. So I'll always stay curious and do what you love and love what you do, and being fearless and expanding the horizons, and keep in mind, you never walk alone. People are there for you to support. And what we learn about health and disease, mostly it comes from like the how we interact with the patients, and we have to need to listen them more to understand more the clinical history of the patients. So I always encourage like the listen more, and actually the patients are telling you the problem, telling you the diagnostic ideas, the present with them. So you never know until you try. And also it's very important to keep in mind that the clinical outcomes are very important as compared to the just only mechanical parameters alone. So thank you for having me today. And I would like to thank the Seungil Row lab members. And it's all about the teamwork. And thank to the Professor Seungil Row and Ken Sanders for all the great supports, and funding agency, particularly the NIH and Rasbu Therapeutics, and our collaborators like from Stanford, Flinders University from Australia, Hong Kong from South Korea, and from SGPGI Lucknow, and for all the supports and clinical insights. So love to discuss the questions you have, and thank you so much.
Thank you. Thank you so much for the talk. So does anyone have questions?
I have a question.